Good afternoon. My name is Jeremy Stool, and I have the privilege of being your host today. And welcome to today's webinar, Animal Models for Neurodegenerative Diseases, an Introduction to AD and PD Mouse Models, presented by Dr. Zhiying Li. Dr. Li is a neuroscientist by training. Her PhD study focused on synaptic transmission and presynaptic vesicle recycling, which was conducted in the lab of Dr. Venkatesh N. Murthy at Harvard University. She did her postdoctoral training in Dr. Jeffrey Friedman's lab at Rockefeller University, where she studied cell type specific hypothalamic networks of metabolic regulation in the context of leptin signaling. Afterwards, Dr. Li joined Taconic Biosciences, focusing on genetically engineered rodent models and for health and disease, supporting genetic science and compliance, as well as scientific commercial production. She joined GemPharmatech US as Director of Life Science and Technology in October 2021. Please feel free to ask questions during the webinar using the Q&A button. Also, this webinar is being recorded and will be accessible from the GemPharmatech website. Without further ado, Dr. Li, the floor is yours.
Well, thank you, Jeremy. Thank you so much for the introduction and being my host today. And welcome, everybody. So my pleasure to share something about the mouse models that GemPharmatech is developing for Alzheimer's and Parkinson's diseases. So I'll start sharing my screen. Okay, great. So right, here's the title. We'll be talking about ADs and PDs. There's a little disclaimer. So I'll be organizing today's chat with you in 4 sessions. Give you a general overview of how GemPharmatech is thinking about developing models for neurodegenerative diseases overall, our one FAD4T for the AD, and a couple of upcoming models in the PD field, and then finally some information about the platforms that we have or services that we have for neurodegeneration or neuroscience in general. So here is the several areas of neurodegenerative diseases that we are thinking about, from Huntington's to frontal temporal dementia, ALS, as well as AD and PD. As alluded to, I'll be focusing on the AD, which is predominantly the pyramidal cells being affected, and in the cortical and the hippocampal areas. And then in the PD perspective is the dopamine neurons that are affected in the dorsal motor nucleus of the vagus or the substantia nigra area. So talking about neurodegeneration or the mechanisms that leads to neurodegeneration, one of the mechanisms or hypotheses that has been prevailing in the field is protein aggregation that came due to the misfolding of pathogenic proteins. The mechanisms that leading to misfolding or aggregation is not fully understood in the different perspectives or different proteins. However, they seem to have a common causal effect in leading to 3 phenotypes or pathogenesis. One of them is a loss of function of the native protein. The second one is a gain of toxicity due to the misfolding or the aggregation. And then finally, the aggregation seemed to be able to lead to inflammation in the nervous system and then leading to neuronal death in the end. So in this slide, what is summarized together is quite a few proteins that have this property. And then the commonality here is somehow this misfolded protein, they can self-propagate and leading to neuronal loss in the patients. So in addition to the misfolding and aggregation of proteins, there are several other common pathways that also taking an effect leading to neurodegeneration. One of them is the autophagy lysosome pathway, and parallel to that, mitochondrial homeostasis can also be perturbed and leading to neuronal dysfunction. And finally, synaptic toxicity or a network dysfunction can also be the cause for neurodegeneration. So with this knowledge from the scientific field in the past several decades, at GemPharmatech, what we want to do is using those knowledge or the understanding of how genetic factors can lead to neurodegeneration to generate mouse models as tools for the study and research, as well as drug development to treat relevant diseases. So on this particular slide, I listed for you how we are thinking to use genetically engineered mice as a tool to mimic these disease phenotypes in mice. We can do KI, which includes the point mutation. We can also do KO, CKO. We can introduce human transgene or mouse transgene, overexpression, targeted expression. We can also do humanization of the mice. And also highlighted for you is the two pipeline platforms, which is KOAP and DSMP. The KOAP represents knockout all project. In this particular project, we are knocking out the coding genes in the mouse genome in a systematic manner, so that for everybody who is trying to study mouse gene in order to understand the protein function or the gene function, this is a great platform. You can basically search, and we have generated almost half of the genome with a KO and a CKO allele for that gene. The DSMP is the drug screening mouse project. What this project mean to do is to introduce human genes into the mouse. The human gene can be healthy allele or disease allele. So with this perspective, we're trying to make the mouse as a screening tool for people, or especially for drug developers, to use these mice to screen the testing articles and to see whether the article has efficacy or safety profiles that are desired for the goal of the development. All right, so with this said, these are the several models that we are trying to develop for the 5 neurodegenerative diseases. And as highlighted in red for you is one particular model in AD and 2 particular models for PD that I'll be providing more information about. And this is the same strain, several list, the list of strains, but I just put on the availability status of this strain. We have several of them that are available already in the red, and some down here in black, and then almost half of them are still upcoming later this year or early 2023. So now let me go to the AD model. So I don't have to say too much about this. So the Alzheimer's disease are characterized by 2 major phenotypic characterizations, A-beta amyloid and tau tangles. Mostly thanks to the work by Dr. Alzheimer and his patient, Auguste D. Detter. And in the past decades and almost a centuries of work, the population of Alzheimer's patients has been diagnosed substantially more and more. And then as our population is enjoying the health benefit of better healthcare, our aging population is also increasing. And as we have known by now, Alzheimer's is an age-related disease. So the older the patient, the more likely we may have Alzheimer's diseases. So millions of people are suffering from it. Not only the population is increasing, the healthcare that has been put into caring for patients with Alzheimer's are also increasing. So accompany to that, or how as a society we can help patients to better deal with Alzheimer's diseases. There are 143 agents that are being tested in 172 clinical trials just in the year of 2022. So this shows how much effort the scientific community as well as the industry, healthcare industry, has been putting into help and developing treatment for this particular disease. On the right side, as you can see, in addition to the very prevalent prevailing tau and amyloid hypothesis, there are also other mechanisms of action that these potential disease-modifying agents can do to help Alzheimer's. For example, the synaptic plasticity or neuroprotection action, or agents that can alleviate inflammation in the patients. Here's just a summary of up to now, the 6 FDA-approved agents that for AD. 4 of them are for acetylcholine esterase inhibitor. One of them is an NMDA receptor antagonist. And then finally, the monoclonal antibody that was conditionally approved for beta amyloid. So going back a little bit, how we can do genetic editing in mice in order to help understand Alzheimer's disease or develop treatment for Alzheimer's disease. So this graph summarizes very well the GWAS studies that has been carried out among human patients. What are the genes that may have an implication for developing Alzheimer's? So as you can see, there's a dichotomy here. On the left side, on the top, there are several genes. There are high-risk alleles for causing Alzheimer's. However, they are very rare among human patients. And then on the opposite side, there are a lot of other genetic alterations in the genome. However, their chance of causing the disease is much lower than the genes that are in yellow. So with this idea in mind, the first thing that we went out to do is to develop a model which is based on the amyloid hypothesis. So here is a summary slide for how the APP protein can lead to the pathogenic A-beta amyloid protein. On the left part is the biochemistry of APP protein. The APP itself can be cleaved in different ways. One of the ways on the right side, it is not leading to pathogenesis of Alzheimer's. However, for some reason, if the protein is cleaved by beta secretase and then followed by gamma secretase, there's a production and accumulation of A-beta 40 as well as A-beta 42. And the accumulation of these peptides will lead to amyloid plaques in the central nervous system. So as important as the A-beta protein itself is the gamma secretase complex that is responsible for doing the cleavage of the protein. And with all the studies so far, there has been a few point mutation identified on the presenilin 1 protein. In particular, the several here listed, they have been implicated in having a role in beta amyloid accumulation. On the right side, vertically is representing the APP gene. And then here, again, is several point mutations that has been identified to have a role in increasing, in particular, the A-beta accumulation. So highlight two of them, or two sets for you. The one on the top is a Swedish mutation, is a pair of PMs that has been shown to increase the chance of A-beta accumulation a lot significantly. And then on the bottom is three mutations, the Florida, London, and the Indiana. You'll see the reason why I highlight those three momentarily. So this is the model that we have developed as a Alzheimer's model, especially focusing on the beta amyloid hypothesis. So in this particular model, what we introduced is the APP, human APP gene, which has the Swedish mutation as well as the Indiana mutation. Parallel to that, we also introduced the human presenilin 1 mutation, presenilin 1 mutant allele, which has these 2 point mutations. So with these 2 transgenes, what we want to do is to introduce the beta amyloid into the mouse. And without using the Florida and London mutation, we're trying to use the Indiana mutation to tame down the beta amyloid accumulation a bit more. So one of the highlights of our model is that we generate this model independently. So we own this model. We want to have this strain available to all users. So there's no constraint for anybody to use it. Here are the data that is summarizing what we see in this model. These are the images from the brain, cortex on the top, hippocampus on the middle, and then several other images to show the plaque in the blown up image. As you can see, beta amyloid accumulation in these cortical or higher brain regions increases as the mice age. which is consistent with Alzheimer's disease and also from other mouse strains that has been reported for beta amyloid. Here is a quantification of the plaque by the area of each one of them. And in terms of A beta 40 and A beta 42 accumulation, these are the quantification of the protein from cortical regions as well as hippocampal regions. As you can see, from either one of those is also following an age-dependent increase. And finally, our collaborator did a comparison between our mice, the F84T, versus the most popular strain, 5X FAD. At 4 months of age, what we see is that the beta accumulation between the two strains are very comparable. So in addition to A beta or the beta amyloid accumulation, we also observe glial activated reactive microglia cells as well as astrocytes in the cortex and the hippocampus. So as shown to you on the left side are the cortical and hippocampal regions with astrocytes. These astrocytes are stained by GFAP. And then on the right side is microglia cells stained by IBA1. And again, cortical regions on the top and hippocampal region at the bottom. And then the row on the top is the wild type animal, and then the lower row is the F84T. So for both cell types, there is a distinct and obvious increase of these two cell types in the cortical and hippocampal regions. And here is a quantification by cell number. And finally, we also looked into neuronal loss as a hallmark of neurodegeneration. So in 8-month-old and 10-month-old animals, what we are seeing is at the cortical, hippocampal, and subiculum areas, we are seeing neuronal loss. These stain are neurons staining, which is a pan-neuronal marker that we can use to quantify the population or the total number of neuronal cells. The quantification is in the works. At the same time, we also used PSE95 Western blot to show when mice are 8 to 10 months of age, we can also tell there's a synaptic loss as indicated by PS95 decrease. So overall, up to this point, at the molecular and cellular level, what I've shared with you is in the FAD14 mice, we see a distinct A beta accumulation starting from 1.5 to 2 months of age. The accumulation of A beta 40 and 42 are age dependent. The older the animal, the more of the accumulation. We can see reactive microglia starting from 2 months of age and very prominent in the 4 months old FAD14 mice. And finally, at older 8 to 10 months old animals, we're seeing neuronal loss as well as synaptic loss. So parallel or in addition to the morphological changes, we have also done some behavioral analysis to see whether these animals are showing some cognitive defects. So the first one we did is open field analysis. Showing to you is 8 months old animal, females on the left, male on the right. What we quantified is comparing wild type and FAD14 mice when they are running around in the open field. The two quantification is the trajectory or the total distance that run by the animal, or the number of entries into the center field of the open field. So as you can tell, there's a trend for females to have more, to be present more in the center, which is more prominent in the male. However, for both animals, there's no no difference in terms of the distance traveled between the two genotypes. And then in the Y maze analysis, what we're trying to show is whether the animal can remember where it has been prior to entering to the second arm of the Y maze. So as you can tell, among the females, the FAD mice seem to remember less where it was. And then there's a trend in the male, but it's not different between the FAD versus the wild type. And finally, this is the Morris water maze, which is a way to test learning memory. And in this particular slide, I'm showing you again is female on the left and male on the right. Out of the 4 panels, the first one on the left upper side is the training, when animals are learning where to find the hidden platform in order to escape from the water. So during that phase, as you can tell, the FAD female mice, they have delayed escape, meaning it takes them longer in order to find where the hidden platform is. And then the other 3 panels are during the trial period. As you can tell from the upper right, the number of crossings these animals have across the probe quadrant, which is the second quadrant, is fewer than the wild type animal, meaning they don't remember. And then the second one here, they cross compared to the wild type animal, which remembers where it should go in order to escape. The FAD animals doesn't seem to remember where is the better route to escape from the water. And finally, it took them longer to find the hidden platform. And all these phenotypic difference is less prominent among the male animals. So we think in our FAD mice, somehow there's a sex dimorphism. The females are more prominent in terms of the Alzheimer phenotypes versus the male. This is actually consistent with human patients. And finally, this is a data slide from a collaborator where the goal is to develop a potential treatment for Alzheimer's. I'll just ask you to focus on the first plot on the upper left side. So as you can tell, there's the control B6 animal, and then the FAD mice in green without the treatment, and then followed by 3 drug treatment group at different doses. For some reason, if you take, for example, the BE panel, for some reason the low dose seem to have the best effect in terms of reversing the FAD phenotype or the Alzheimer phenotype compared to the middle dose or the high dose. So roughly speaking, it is probably a tool for our collaborator to use these mice to screen their drug. Parallel to that, we think FAD mice is ready to be used as a screening tool for other potential reagents. All right, let me see. Okay, so here it comes to almost the end of the FAD mice. I would like to summarize what we have seen so far from these mice, and then comparing that to 2 major mouse models has been widely used in the AD field. So as you can tell by the color-coded bars, in the FAD40 mice, the A-beta accumulation, the plaques, as well as the appearance of reactive microglia, in our mice, these 2 phenotypes seem to have an early onset compared to the 5X FAD or the 3X TG. Parallel to that, though, the cognitive impairment or the loss of neuronal cells seem to have a delayed onset. So we're hoping, this window from the beta amyloid until the cognitive defects or the behavior defects is opening up a longer testing period for people who want to use these mice to test the efficacy of a potential treatment. So these are just words to summarize what I just shared with you. Now, as you know, A-beta presenilin, these are only the 2 high-risk genes. Although they are high risk, they are low prevalence among the human population. So what's the better way, or what's the alternative way that we can develop preclinical mouse models to help with research as well as drug development? Here is what we are calling the AD+ mouse models platform comes in. So as you remember from before, there's a lot of other genes or risk alleles that has been implicated in Alzheimer's diseases. What we want to do is to introduce this implicated human genes into the mouse so that the human protein can become available as targeting tools for testing. So with that as our goal, we have initiated several strains where we're introducing wild-type human alleles or some mutant alleles into the mouse. So this is the current status of that AD plus mouse model project. Right. As we wrap up the AD, I'm going to give you a little introduction into the two PD models we have been working on. So Parkinson's in the ballpark is a neurodegeneration in the motor function. And people with the disease tend to have slowness in motion as well as imbalance and tremors that are uncontrolled by the patient. And here, with the interest of time, I'm not going to go through the details of this, but it's just a lot of the pathology as well as pathways has been implicated for Parkinson's diseases. And jump out to my eyes are this alpha-synuclein and mitochondria. So there's another way to summarize the alpha-synuclein hypothesis and how that is leading to the PD phenotypes. In our hands, the first model we tried to develop is the alpha-synuclein model. And here we use the back transgenic approach, introducing a human alpha-synuclein cDNA. And in this particular allele, we have the A53T mutation. So this mutation seemed to lead to the alpha-synuclein accumulation in Lewy bodies. So showing you on the right side is the validation. So we're doing Western to see the human protein is present from the mouse brain. At the cellular level, what we can see is starting from 8 weeks of age, we can see the protein accumulation in the cell in the midbrain, and it progresses as the mouse age. So up to now, we are assaying animals at about 6 months of age. Parallel to the accumulation, we're also seeing that at the substantia nigra area, we can start to see neuronal cell loss. For example, at 12 weeks of age, as you can see on the top is the wild type animal. That region of the cells are lost in the transgenic animal. And then at 16 weeks of age, the loss is more pronounced than the wild type animal, or compared to the wild type animal, and more cell loss compared to the 12 weeks animals. So a few simple behavioral testing using these mice. So on the left side is the grip strength, showing the muscle strength of the animal. We have been seeing a female and male. They seem to have lost muscle strength when they are 12 to 18 weeks of age. And then on the lateral testing, they don't seem to have a very profound imbalance or loss of endurance yet. However, the trend is there among 18 weeks old animals. And finally, parallel to these behavior changes, we do see a change of body weight between the mutant versus wild type. So here again, using the same type diagram, summarizing what we have seen from our back transgenic animals, they seem to have the dopamine deficiency as well as the motor imbalance early on or earlier than other alpha-synuclein pre-mutant animals. At the same time, the alpha-synuclein inclusion in Lewy bodies also seem to have an early onset, and additional phenotypic analysis is still ongoing as we speak. And finally, alpha-synuclein is one of the hypotheses that leads to dopaminergic cell death. Another one is mitochondria homeostasis. So there has been several papers talking about the TFAM gene and its activity or implication in Parkinson's diseases. And on top of that, there has been a paper published about the mitopark mice in a PNAS paper published in 2007 by Dr. Mats Ekstrand and Lindquist from the Karolinska Institute. And in that seminal paper, the mitochondrial homeostasis and how that can lead to Parkinsonism is very well characterized. So in our hands, with full credits to the papers that has been published in the field, we also generated the TFM conditional knockouts as well as the DAT-free animal. So in our hands, we have crossed these two strains together to generate the TFM homozygous flox allele as well as the DAT-free positive animal. So showing you here on the right is proof of principle, as you can tell, between the wild type animal on the left versus the double mutant animal on the right side, there is profound neuronal cell loss in the substantia nigra area in both 10-week-old and 14-week-old animals. And then we can see some body weight change due to the mutation, which is not so surprising because mitochondrial homeostasis is very important for metabolism. And on the behavioral side, the grip strength and the rotor rod tests seem to show that these animals do have motor function defects. In particular, the rotor rod test has been very profound for both males and females at the age that are indicated in the plots. So finally, with the next couple of minutes, I'm going to do a little introduction of the platforms that we have or the services that we can do for customers who are working in neural degeneration or neural science in general. So here, as I have shown you results from the several behavioral testing platforms that we have, we can do open field, grip strength, rotor rod, gait, treadmill, which I didn't show here. We can also do novel objects, Y maze, as well as Morris water maze. In addition to those, we also do forced swimming, sucrose preference, elevated plasmase, as well as tail suspension. So beyond this behavioral testing, as I shared with you, GemPharmatech is very strong in terms of generating new genetically engineered mouse models. So with every new mouse model we generate, we also support the phenotypic characterization of the new strain with our phenotyping platform. Within this phenotyping platform, what we can do is validating the DNA where the mutation is supposed to take place, as well as RNA protein, and all the way until cellular analysis, as well as tissue sectioning, using HSE, H&E staining, as well as IHC, in order to tell or characterize how the mutation has been leading to cellular or tissue level changes. And then once we verify that, what we can also do is functional analysis of the mutation in the function of the whole body of the organ or of the living mice. So here, I have shown you the behavior analysis platforms we can do. We also do metabolic analysis. Prominent to that is, DEXA for body composition and metabolic cages for O2, CO2, and heat and ambient activity. In the metabolic field perspective, we do glucose homeostasis, insulin sensitivity or tolerance tests, and lipid metabolism from serum or from liver. And we can also do a full panel of blood analysis from living mice. So after that, with the most recent advance in the immuno-oncology field, we have a full spectrum of flow cytometry that we can use to assay peripheral immune cell types. And we have an in vivo imaging platform, ARIS, to look at wherever the luciferase is tagged. So in the IO field, a lot of times this platform is used for tumor analysis. And then in other metabolic fields, for example, fat has been tagged or can be tagged with luciferase in order to enable in vivo imaging of fat tissue composition. All right, with that, I'm going to close the webinar or the presentation, and we'll be open for questions.
What's the advantages of FAD4T mice?
Well, if that's in the context of in comparison with the 5X FAD or any other strains, we think our mice is, having an early onset of beta amyloid, so is a, very distinctive parameter that people can measure of the start of accumulation of beta amyloids, but it has a later onset of cognitive or behavioral deficits, which means in between there's a longer time window for people to test whether a potential therapy can actually reverse the beta amyloid accumulation and delay the behavior or cognitive deficits. So we're hoping this really becomes a screening tool for the drug industry in order to help more patients who are having the disease or will develop the disease.
Thank you. And then it looks like there's one final question here as we wrap up. Are you planning to test some traditional drug efficacy on your PD and ALS models?
Internally, not really at the moment. We provide services, and then in our service packages, typically we will have positive control and negative control. So very well, also pertaining to the customer's desire, which drug that we will use as a positive control for the test service will be decided based on the project itself.
Great, thank you. I'm not seeing any other questions that are coming in at this point. We are kind of long on time. Is there anything else you'd like to share, Zhiying, before we wrap this up?
Not really. We presented a poster on the FAD mice at the current Society of Neuroscience annual meeting in San Diego. So if you are SFN, I encourage you to visit GemPharmatech at booth 2500. You are more than welcome to visit the website or contact us if you have more questions or you want to have more information.
Great, thank you. And again, thanks for such an informative presentation today. And thank you everyone for joining us. We look forward to seeing you at our next webinar in December. And as Zhiying said, please check back on our website for updates coming for what that topic will be. And if you have any questions, feel free to visit our website again and fill out a contact us form, and then we'll get back to you.
Yep. Thanks, Jeremy. Thank
you, everyone. And I'm going to go ahead and close things out. Have a great day.
You too.
Bye-bye.
Bye.